We are going to learn today about synaptic plasticity. So in the previous lecture, we had studied about the properties of synapses, and I think we have covered a big chunk of properties in our previous class. And continuation to that, we are going to study about synaptic plasticity. This is also a property of the synapses. Now, what is the first thing that comes into our mind is what does the word plasticity means? So plasticity in general term means the capacity to change. Okay. And when I tell about that synaptic plasticity, it means the capacity of the nervous system to change. So that is what we refer to as the synaptic plasticity. Now this change, how long is going to stay? Is it going to be a short term or long term? It can be in both the ways. It can be a short term, which can be present, or you can say it can stay on for minutes to seconds, or it can be a long term where it may continue for minutes to lifelong. Where does it occur? Obviously, the name suggests it is a synaptic plasticity. So it is occurring at the synapses. And it is no use of telling all of you here that synapse is nothing but what is a synapse? Synapse is a junction between the two neurons. So plasticity being a feature of synapse gives a scope for the synapse to arborize. You can say you can increase the number of synapses in the central nervous system, or it can even decrease. So it affects the structure and function of the neural circuits and systems, and it can also affect the organization of the functional representations, like somewhat called as the cortical mapping. In some instances, we can take an example as if we do not use any part of our body, although it has a cortical mapping, if we do not use it for a long time, obviously the areas which have been allocated for that particular part may be having less number of synapses. So there are different experimental proofs about it. We will not go into detail about. Now we'll go into the mechanism of it. Before we go into the mechanism, we should know that the synaptic plasticity, it forms the basis of memory. Okay. So whatever we try to learn, we kind of form a memory of it. And this memory again has a broad classification. We can have short memory. Short-term memory can be termed, consolidated into long-term memory. Now, formation of memory, the cellular basis of formation of memory, it involves synaptic plasticity. Then acquisition of motor skills. You want to learn something new. For example, riding a bicycle or driving a car or something like that. What you do? Initially, you try to learn about it, then you practice it, you habituate yourself, and then you become acquainted to it. So kind of it is like you're acquiring the knowledge, you're applying the knowledge, you're repetitively applying the knowledge, and then a stage comes where you have acquired the knowledge. Then acquisition of cognitive skills like learning, language, and adaptation to recovery from injury, or it can be from any disability. Now, when we talk of plasticity, I think this is the one that your brain thinks, it has a two-way. Either I can just do nothing, wait for the time, and take a route towards aging, where I'll be losing all my arborization, I may not have more of a growth or a development to that. Or I myself go towards the plasticity. Means I take up a new thing, I learn about it, I rehearse it, and I make new synapses. So plasticity is a dynamic state of the central nervous system. The most important thing that, before we go into the types of plasticity or the types of synapses and all that, we should know what is the core of the mechanism. What is the core mechanism of plasticity? Since it is involving the synapses is the junction between the two neurons, and they kind of interact with each other. We have a presynaptic terminal or a presynaptic element, which is communicating with the postsynaptic terminal. And in case of chemical synapse, there is a neurotransmitter which is involved in it. So it is kind of, the whole of synaptic transmission will take place, but for plasticity, there is certain mechanisms which is kind of attributed only to plasticity. And what is that? The neural activity, it triggers the activation of postsynaptic second messenger system. We are going to learn about it. We'll know what the mechanism is. So one thing is there, your presynaptic is going to change the postsynaptic environment. Okay. And that environment change can be through the second messenger system. Then it will trigger usually an alteration in the level of intracellular calcium. Calcium plays a very important role in plasticity, especially, how much of a calcium is available, what is the time taken for the reuptake of the calcium, or you can say recovery of calcium, that also will play a role in plasticity. So trigger is going to do what the trigger is going to do. I'm going to get a trigger from the presynaptic end, and that trigger is going to change my postsynaptic environment. And the postsynaptic environment is going to respond by the second messenger system, where we can have the calcium dependent second messenger system, which is going to alter the activity of protein kinases and phosphatases. We are going to go into detail about it, and I'll show how it is changing and how our plasticity is going to get changed. Then there can be a long-term change, which can be due to alteration in the protein phosphorylation, which is going to mediate, the early stages of long-term synaptic plasticity. So this is in general, what are the different cellular mechanisms involved in synaptic plasticity. Now, the first thing we should know is what are the forms of synaptic plasticity. Generally, there are two forms of plasticity. One is called as the intrinsic or the homosynaptic mechanism. Other is called as the extrinsic or the heterosynaptic plasticity. Homosynaptic mechanism is like where it's like feed-forward system, where one presynaptic is involved with one postsynaptic. And here it refers to the changes in the strength of a synapse that are brought about by its own activity. Okay, so the synapse itself is having some activity which is changing the plasticity in other way. So let me just take you a kind of, okay, I do have slides in the upcoming slides where we can see what is a homosynaptic mechanism and the heterosynaptic plasticity. So homosynaptic in one word, it can be something like this. Okay, let me take a pen and show you. So it can be something like, okay, let me see where my pen is. Okay, it's here. Homosynaptic means, okay, this is my presynaptic, this is my postsynaptic element. So whenever there is, I have the signals coming from the presynaptic, which is in the form of neurotransmitters. These neurotransmitters are going to bind to the receptors present in the postsynaptic. And because of these neurotransmitters binding to their receptors, there will be some change in the polarity of the postsynaptic element. Now, these changes that you get in the postsynaptic element is due to this presynaptic activity. So this type of synapses are called as homosynaptic terms. We can use this as homosynapse. Same activity is brought about by the influence of the other presynaptic, which is there itself. Okay. Then what is heterosynaptic? Heterosynaptic is something which I can show you here itself. I take a presynaptic, I take a postsynaptic. Then I have somebody else also. I have someone else who is going to influence my presynaptic. I think this is what we have learned when we were trying to see the properties of presynaptic inhibition. So such type of synapses where I may get an effect, where there is a change in the strength of the synapse, which is not only because of this way, but rather somebody else is influencing. So some other synapse has come to the presynaptic end, and that is influencing the whole activity of this synapse. Such type of arrangements, we call it as extrinsic plasticity or the heterosynaptic synaptic plasticity. So intrinsic means that you're getting the changes in the synapse because of the own activity of the synapse, since synapse is between the two neurons. Okay. So here, this is homosynaptic. When the influence or the activity, there is a change in the activity, not because of the two elements of the same synapse, but rather somebody outside, an outsider has come and who has changed the activity over here. Now coming to the homosynaptic plasticity. Now let us take it very carefully. We'll go on and let me take a laser. Now, let me start from here. If this is visible to all of you, here, what do I see? I see a synapse of a sensory neuron and a motor neuron. And there is a microelectrode, recording electrode inserted into the sensory neuron and electrode inserted into the motor neuron. So this may be like an example of in the spinal cord, we have a synapse like this, where we have 1A fiber coming from the muscle spindle, synapsing onto the alpha motor neuron. So this is that type of arrangement. Okay. Now let us go to this. This is an example of homosynaptic plasticity. And homosynaptic plasticity, they can be, you can say two ways of it. Either there is synaptic facilitation or there is synaptic depression. Now I will take you first to the synaptic depression and then I'll take to the synaptic facilitation. In synaptic depression, let's just first go to the arrangement of this recording. You have taken a stimulus, you have taken a small stimulus. There is a stimulus there in the sensory neuron and an EPSP, or you can say postsynaptic potential, is recorded. One impulse or one stimulus, one EPSP is recorded. Then after a lag of 200 milliseconds, you give another stimulus of the same strength. And what do you get? You get another EPSP. But what is observant between this EPSP and this EPSP? This EPSP is of a smaller height. Now, what has happened at this time? There is something called as synaptic depression has happened. We'll talk about the mechanism later, but now all that you have to see is observe the height of the EPSP. Here, you gave an impulse, you got an EPSP. After some time, when you gave another impulse, you got an EPSP. But what has happened here is that there is a decrease in the height of the EPSP. Now, what is the mechanism behind this? When you gave an impulse, when there was an impulse coming from the sensory neuron, there was release of neurotransmitters. When there was a release of a neurotransmitter, so with one impulse, suppose I got a release of 10% of the neurotransmitter, which was present at the presynaptic end. Okay, I think for this, we should go to the white screen. That will be easier for us to understand. Let me take you to a white screen. Fine. So we'll go back again and take a pen. Okay. So what do? I draw a synapse first. Fine. Suppose this is my excitatory synapse. This is my sensory neuron. And this is my motor neuron. Fine. Good enough. What do I do next? Okay. In the sensory neuron, it's my presynaptic terminal. I have the vesicles. Okay. And these vesicles are filled with the neurotransmitters. Okay. Good enough. Now, what's the next thing that we have to do? We have to again start with the steps which are involved in synaptic transmission. What is the first step? There is arrival of the action potential at the nerve terminal. Because of the arrival of the action potential at the nerve terminal, there has been a change in the potential or the polarity at the presynaptic end. And that has led to the opening of voltage gated calcium ion channels. Okay. Once this channel is opened up, ECF calcium being more than the ICF, there is influx of calcium ions. Calcium is the main factor here. Okay. When there is availability of calcium ions, these calcium ions are going to bind with the anchoring proteins of the vesicles. And these vesicles, what are they going to do? They are going to come near the presynaptic terminal end. Oh, I just missed it. Fine. So these are the vesicles filled with the neurotransmitters. Once there is arrival of the action potential, there is influx of the calcium ions. The calcium ions, they have an important role. They are actually, what are they going to do? They're going to bind with the anchoring proteins present on the vesicles. Along with these membranes, they will act as a bridge for these anchoring proteins. These vesicles will attach to the presynaptic end and they will release the neurotransmitter. Okay. So when I gave one stimulus, I got one EPSP. Okay. So what did this happen? These receptors, the neurotransmitters, they came and they bound to their receptors. These receptors again are of two types. One is called as ionotropic type of receptors or ligand-gated channels, you can say. Ionotropic, straightforward receptors who will act like ion channels. Or there can be something called as metabotropic ion channels. What is the difference between ionotropic and metabotropic? An ionotropic may be something like this, okay, a direct channel. When the neurotransmitter comes and binds to it, there is some confrontational changes and these will act like ion channel and they'll open up. Once they open up, they allow the influx of cations, especially the sodium or calcium, whatever is present. How about the metabotropic ion channels? Metabotropic ion channels are also ion channels who will have the site for the neurotransmitters, but they will be linked with some G-coupled protein, okay? And these G-coupled proteins on activation will bring about certain changes in the cell over here, the postsynaptic element. And that is going to bring about the changes. What are those changes? They can either directly go and open up the ion channels, other ion channels, or they may cause some transcriptional changes. And because of that, there will be production of more ion channels. And these ion channels may go and attach to them to the postsynaptic end and hence will cause the increase in the influx of the ions. So coming, after knowing that these are the two differences, let me take a clear screen now. So I hope we have understood that there are two types of ion channel receptor proteins. And what are those? One is ionotropic, other is metabotropic. So when I'm trying to explain you about the synaptic depression, let me take you again to that same diagram where we form a synapse. And what has happened? What was that? That we gave one impulse in the sensory neuron. We gave one impulse. And what did we record in the motor neuron? We had recorded a EPSP. Okay, fine, well enough. So first step, there were steps of synaptic transmission. There was release of neurotransmitter. Neurotransmitters went and got bound to their receptors. And as they bound to their receptors, there was a small change in the polarity and we got a EPSP. After a gap of 200 milliseconds, I gave another impulse. I gave another stimulus. And what did I get? I get a smaller EPSP. Now, this is what you called as synaptic depression. What is the reason? Let me come back to you. So during the synaptic transmission, the steps were like, once the action potential has arisen, there was influx of calcium ions. Okay. So this impulse, first impulse, had caused an influx of calcium ions, and these calcium ions had caused kind of release of neurotransmitter. Suppose this release was the 10% of the total vesicles total number of neurotransmitters present, right? So once this 10% was released after the first impulse, we gave some time, but not a very long time, but we gave some time. After some time, there was a second impulse which came to the presynaptic end. And what did it do? It has now released 10 other percent of the remaining 90%. I hope you make it a sense. I make it a sense of it. What does this mean? So take an example. I was having 100 neurotransmitters. When I gave the first impulse, I got only 10 of them released. Okay. And since this second impulse is coming after after a gap of 200 milliseconds, what has happened? What am I remaining now with? I'm having, suppose, remaining with 90 neurotransmitters. When my second impulse came, second impulse is also causing, about 10 of the neurotransmitters to be released. But this 10 is released from the 90. Okay. So what has decreased? It has decreased the amount of availability of neurotransmitters. When your first impulse had come, you had 100 of them available. So you could release 10% of that 100. Then when you release that 10% of that 100, you got an EPSP at the postsynaptic terminal. Then after a small gap, not so big gap, but a small gap of 200 milliseconds, when you gave another impulse, you kind of already taken 10% of the previous one. Now you are going to take 10% of the remaining 90%. Got my point? So that is the main mechanism for the synaptic depression. This is why you get a smaller height of EPSP. Now, what is synaptic facilitation? Synaptic facilitation is let's first see how we get this synaptic facilitation. Suppose we have given the first impulse. When we gave the first impulse, we got a response. Okay, fine. Then we gave a small gap of 20 milliseconds. Then we gave another impulse. Here, what is the factor that you have to see? What is the difference here and here? In both the cases, the strength of the stimulus is same. But what is different is the timing. Timing is a factor which is changing both in synaptic depression and synaptic facilitation. How this facilitation has been brought about? Let's see here. What has happened here is this, for this situation, again, let me take you to a new white screen. Fine. So we'll draw back again one synapse, another synapse. Fine. What did I do? I gave the first impulse. Okay. First impulse came. There was a release. Once there was a release, there was binding. And what did I get? A postsynaptic impulse like this. Fine. Then immediately after 20 seconds, I gave my second impulse. When I gave my second impulse, I got a increase in the height of my EPSP. Well, what is this mechanism here? What is it showing here? This is showing something called as temporal summation. And what is this temporal summation? Temporal summation is the property of synapse where the two EPSPs, they get summated. And how do they get summated? The situation is that this is a presynaptic neuron. This presynaptic neuron is bombarding impulses one after another within a short period of time. This timing is very short. So what has happened? There has been a summation effect. Since EPSP is a local potential and there is a property of EPSP that it can be summated. 2 EPSP or 2 strength can be summated. Now, let us go to the mechanism of it. Okay, so what is the mechanism? Again, the same thing, calcium ions. Availability of calcium ions. Okay, availability of calcium ions. How? Let me tell you again. So what did it happen? Let us go back to the steps of synaptic transmission. There was an impulse came, okay. Then there was influx of calcium ions, fine. These calcium ions cause the release of neurotransmitters. Good enough. These neurotransmitters came and bound to their receptors, and these receptors, thinking they are the direct channels, ionotropic type of receptors, there was a propagation of EPSP. Not a propagation, sorry, there was a generation of EPSP, a local potential. Then, within a short period of time, I had another impulse coming. Right? What did this impulse do? This impulse actually has caused the more availability of, it has added up more number of calcium ions. How did it all happen? Let me tell you. First impulse, suppose, caused the entry of 3 calcium ions. I'm just taking an arbitrary number of 3 calcium ions. With one impulse, I'll get 3 calcium ions. But after, these 3 calcium ions got in, they caused the release of neurotransmitters. Neurotransmitters went on and binding and all that has happened. That's an after stimulation, the effects are happening. In the meantime, what is the fate of my calcium ions? Calcium ions, they go back to their own sources. Okay. Maybe they go back to where they have come from. As there is this change in the potential, they will go back where they have come from. So there is something called as recovery or, reuptake of calcium ions or recovery of calcium ions. Now, this recovery also takes some time, right? Suppose when my second stimulus came, I already had one calcium left over from the previous one, which has not been completely reuptaken. And my second stimulus is giving me 3 more calcium ions. Now, in total, how many calcium ions do I have? 4 calcium ions. So there is an increase in the availability of calcium ions. And since neurotransmitter release depends on to the concentration of calcium ions, your EPSP, which will come, the impulse that there will be a traveling of these impulses, they'll come here. What will happen to my response? That will get a temporal summation. There will be an increase in it. Okay. So these temporal summation that you're getting, because of that, there is increase in the height of EPSP. So that is what is called as synaptic facilitation. I think I should just tell you. Now, isn't it so funny? In one place you are telling me, I think this question will come in both of our minds. What is that? In one place you are telling this is because you gave 2 impulses. You gave a second impulse and there was a decrease in the height. Here you gave me second impulse, but there was an increase in the height. Height is showing that, look at it. It is 1 millivolt here and there is an increase to 2 millivolt. Height of contraction, height of the, not contraction, height of the EPSP has increased. Now, how this has happened? Again, we'll go to the differentiation. The difference over both of these for depression and facilitation is the timing. Timing of the impulse. Here you gave enough time for the calciums to be reuptaken to go for their recovery. Here the time was not given. So there was an availability of calcium ions from the previous impulse for the second impulse to utilize it. So there was kind of facilitation or beneficial effect. This is also sometimes termed as the beneficial effect. And because of that facilitation, you get an increase in the height of impulse. There is an increase in the potential. Then, yes, this is what I have been telling you. So these are the 2 graphs, which is going to explain you the first phenomena. The first phenomena is synaptic depression. So what has happened during depression? When you gave the initial, you got the transmitter released was like this. Okay. Then what has happened? This was my basal calcium ion concentration. But when you gave the impulse, when there was an impulse, then that has caused the influx of calcium ions. I got a change in the concentration of the calcium ions. This is my initial stage. Then when I gave the second stimulus, what has happened here? What about the neurotransmitter? Your calcium ions, look at this. Even at the peak, there is no change with the concentration of calcium ions. That is not the main factor, but the main factor was the availability of the neurotransmitters. The neurotransmitters were less available. And so what did I get? A depression. Okay, so this is what is called as synaptic depression. How about facilitation? What happened? I gave the first spike. There was a first impulse that gave me, this is the release of the neurotransmitter because of the first impulse. Immediately, we gave the second impulse. What has happened after these initial impulse? There was a recovery of calcium ions. Okay. After the first impulse, the normal phenomena is that the calcium will try to go back to its own store area, or there will be utilization of that calcium. So there is a recovery phase. But by the time it was recovering, I had given the second stimulus. So this much of residual calcium was still remaining. So who is the main factor for this increase in the transmitter release, which is facilitating my synaptic EPSP? That is the residual calcium. So residual calcium in case of facilitation is the main cause, and decrease in the neurotransmitter release is the main cause for synaptic depression. Now, there is another property of synaptic plasticity, what we call as post-tetanic potentiation. So let us first try to understand this picture. This is a presynaptic end. This is a postsynaptic end. I have the recording electrodes both at the pre and the post. So what are the recordings? So this is the presynaptic action potential and this is the postsynaptic action potential. Let us go one by one. First, you gave the impulse. So there is volley of action potentials, which is coming from the pre. And how is my post responding to these initial action potentials coming from the pre? It is giving my post EPSPs. Okay. Then a time comes when you give a volley of impulses, like a tetanic impulses at a very quick successive intervals, very quickly. You're giving one after successively, one after another, you kept on giving the impulses. What is my response here in the postsynaptic end? I'll get a, what is this? Synaptic facilitation impulse. What do you call as a beneficial effect? You get here is a beneficial effect. With, as on, we kept on giving the impulses, there was an increase in the height of the EPSP. Okay, there was an increase in the height of EPSP. Again, who is the main factor here? The residual calcium ions. Okay, so this was because of the residual calcium ions. Then a situation comes that we again come back to the normal volley, same normal volley of action potentials as the previous one. But what has happened? Here we find that still we find an increase in the height of the EPSP. What does this mean? That my postsynaptic potential, I'm getting a increase in the potentiation post-tetanally also, after the post-tetanus state. So even after the tetanus, volley of the tetanus has stopped, I'm still getting a increased, or you can say a beneficial effect in the postsynaptic end. Okay, this type of effect that you get is called as post-tetanic potentiation. And these post-synaptic potentiation, this is going to continue for longer period of time. And when it continues for long period of time, we call it as long-term potentiation or LTP. Okay. And this LTP, for time being, believe me, and just remember that LTP is the molecular basis for learning. Okay. So how do I illustrate it with what example? So suppose you are learning something. Okay. So if your brain, suppose you do something very small for a small period of time, you do it. As you do it, you are getting, your presynaptic impulses are coming, your postsynaptic element is giving the responses. If you do not do that continuously, you will get something very similar. It will keep on. As you do, you will get a response. As you do, you will get a response. But what if you do the same action again and again, again and again? You repeatedly do it. So what is your brain doing now? How is the synapse responding? Synapse is responding by increasing the EPSP. And this increase in EPSP The molecular basis, although is a beneficial effect, and that is mainly due to the availability of calcium ions, I find a post-tetanic potentiation. Okay, this is a tetanic potentiation. But what is so good about it is, even after I have stopped, what does it mean? That I kept on learning something. I learned it. I tried to memorize it once again. Suppose I say one word 100 times. If I have done that one word for 100 times and I stop that, I do not read that word again, what has happened to my brain? My synapses have gone for an increased long-term potentiation. So next time, the moment I read that word, I immediately remember the spelling of that. And how do I remember? Because there has been a synaptic plasticity. There has been an increase in the potentiation at the same synapse, which I have formed due to repetition of that word. Okay. So hence that is how it forms the basis for memory. Now, that all that we have learned till now, this post-tetanic potentiation and the synaptic plastic facilitation and depression, what was the thing that we were doing? We were doing on a homosynaptic arrangement. Okay. Now let us go to the heterosynaptic plasticity. What is this heterosynaptic plasticity and where will this be present? This is one example to show you the heterosynaptic plasticity. Look at this arrangement. There is a presynaptic neuron or an element. There is a postsynaptic neuron. But there is an involvement of a modulatory neuron. Who is this modulatory neuron? A neuron who's going to modulate the function of my presynaptic impulse. So either it can inhibit my presynaptic neuron or it can excite my presynaptic neuron. So it is kind of modulating the function of my presynaptic neuron. Okay, let us see. So as we have in homosynaptic plasticity, depression, and the facilitation, similarly here also, they will also enjoy the same thing, facilitation and depression. So let us go to the presynaptic inhibition and look at what depression means here. Okay, so there are two situations, a control situation. Control situation is only pre is giving the impulse, the post is receiving it and responding it by giving a response of EPSP. Here, we gave a presynaptic impulse. We responded by a postsynaptic potential. How about the ionic conductance of the calcium ion? Because of the presynaptic response, there was an increase in there was a conductance of the calcium ions. And because of that conductance of calcium ions, there was other activities of the synaptic transmission. Well enough, that is a control condition. Now, when we involve this modulatory neuron, when we activate this modulatory neuron and take into mind that this modulatory neuron is my inhibitory neuron. So what it will do? It will inhibit my presynaptic neuron and it is going to decrease the conductance of calcium ions. How will it inhibit? By decreasing the conductance of calcium ions. So here, look at it. I gave an impulse at the presynaptic end. What do I find? A smaller response. Okay, a smaller response of EPSP. And what is the reason for this? A decrease in the conductance of the calcium ion at the presynaptic end due to the inhibition by this modulatory neuron. So this is an example where we have already learned in the presynaptic inhibition. So that is also, you can fit up here as heterosynaptic plasticity as example of synaptic depression. How about the presynaptic facilitation again here? As I told you, this modulator can be an excitatory or it can be inhibitory to my presynaptic neuron. In the presynaptic inhibition, this was inhibitory. Now, if it is excitatory, after the control examination, a controlled way of impulse transmission, here what has happened? The presynaptic neuron is giving an impulse. The post is giving a response of EPSP. Looking at the conductance of calcium ion, this is how we get it. Suppose I have activated M1. M1 is my modulator neuron. I gave an impulse at the pre. I got a bigger response at the post. And how did it happen? Because this modulatory neuron has increased the conductance of calcium ion in the so availability of calcium ion has increased. And obviously, the strength of my stimulus is dependent on two factors. One is the neurotransmitter availability, other is availability of the calcium ions. So more of calcium is present there, so more amount of neurotransmitter is being released. So this is presynaptic facilitation. So what we have studied till now is homosynaptic plasticity. In the homosynaptic plasticity, we can have synaptic depression or we can have synaptic facilitation. Then we went to heterosynaptic plasticity, where we can have presynaptic inhibition or presynaptic facilitation. Now, let us take an example of a Purkinje cell. Let us take a circuit. We are kind of taking of a circuit, a cerebellar circuit you can take. There is a postsynaptic neuron, and this is the dendrite having the dendritic spines. Or you can say, let us take an example of hippocampus. Hippocampus is such structure in your diencephalon. This is a structure of diencephalon, which is mainly responsible for the spatial memory. So what exactly happens is that all the memory is being formed. They come to the hippocampus. Then hippocampus consolidates that memory, short-term memory into long-term memory, and it sends back to the cortex. And the cortex is the one which is storing your memory. Okay. So let me take an example. So very well, synaptic plasticity is very well studied in the hippocampus. It is also studied in other structures like cerebellum, cerebral cortex, but it is widely studied in the hippocampus. So this is one such example of hippocampal circuit where I'm having the pyramidal cells, and I do have the dendrites and the dendritic spines, and there is these afferent fibers. Now what is happening here? I am having two recording electrodes, one at the postsynaptic end and the other at the afferent fibers end. Now let us see this. So initially, since we have taken the EPSP as 100%, the smaller impulses. So these will start from here. This is stage 1. So let me take my pen and show you. Okay. So this is my first stage. What did I do? I started with a presynaptic stimulus. Okay. So action potential has arrived the presynaptic end, and it is going to give rise to the EPSP, smaller EPSPs. And those EPSPs which has come, we have taken it as 100% of the control, means it is the starting of it. Then you gave the second, this is the second one. Here you started giving lot of successive stimulus. So lot of tetanic type of impulses started coming. What did I get? I got a post-tetanic potentiation. What happened to my EPSP? It has increased, it has increased more than 200% of the control. There is an increase in the height of the EPSP, and that is mainly due to the summation, temporal summation you can call, or special summation. There has been an increase. Okay. Now, even if I have stopped the tetanic, this is stage 3, where I have stopped the tetanic impulses. Number of impulses is back to what the initial I started in stage 1. But what do I see? My EPSP is almost 200 of the control, means there is an increase in the potentiation. It is at a higher potentiation. So it is going to respond very quickly to the impulse which will come from the presynaptic end. And this increase in the EPSP that you get, and which is going to present there for long period of time, we call this as long-term potentiation. LTP stands for long-term potentiation. Fine. So I'll repeat it once again. We start with a situation of control where, so we had, I'll draw it here rather one terminal to show you. Okay. Stage 1, you started giving some impulses. This gave you EPSP. Okay. These were the EPSPs, and that was kind of 100% of the control. Then you increase the number of impulses. You kept on giving many impulses. And what happened here? The EPSP was kind of increased. I found an increase in the EPSP. Then what did I do in the third stage? I stopped this tetanic impulse. I went back to the normal rate of impulse. But what has happened to my potential? It has reached to a increased level of response now. Even after the tetanus, it has gone. It doesn't come back to its original stage of this. Still, it is remaining here for a long period of time. So that means even with a small impulse, I'm going to have a good amount of response. So that is what you call as the long-term potentiation. Now, this is an example, this was actually an animated one, but I have taken it from this site. This is my source here. So let us take a situation before the tetanus. Now, this whole thing, how it is all explained, this long-term, for understanding the long-term potentiation, we have to take into consideration a neurotransmitter glutamate and the receptors of glutamate. The two receptors of glutamate are AMPA receptors and NMDA receptors. Okay, so these are the two receptors of the glutamate, ionotropic receptors of glutamate, AMPA receptors and NMDA receptors. So if my synapse is a glutaminergic synapse, the presynaptic neuron is going to release glutamate. Okay, so once this glutamate is released, this is going to bind. This is my glutamate. This is going to bind to its receptors. And what are those receptors? AMPA receptors and NMDA receptors. Once the glutamate binds to the AMPA receptors are actually an example of ionotropic type of receptors. So what is the feature of ionotropic receptors? Once the receptors are bound to their ligands, they are going to change their configuration and they are going to allow the influx of the ions. And which ion is more abundant outside? It is the sodium ions. So the cation, there will be influx of the cations, especially the sodium ions. Okay. But what about the NMDA receptors? NMDA receptors are also glutamate receptors, but they have little different feature than the AMPA. You know what? They are blocked by magnesium. There is a magnesium ion which is blocking this NMDA receptors. If I want to compare between the conductance of AMPA and NMDA, NMDA is a bigger channel. It can allow more influx of cations as compared to AMPA. But what is the situation here is that this NMDA is blocked by magnesium ions. Okay, so this is my situation before I started giving the tetanic impulse. So let us go to situations. Let us start with the events. So one impulse came. So there was an action potential at the terminal end that caused the release of glutamate. Glutamate came and bound to its receptors, that is the AMPA receptors and NMDA receptors. In NMDA receptors, they couldn't make any change. Why? Because NMDA was blocked. The channel is blocked by magnesium ion. Then only poor AMPA could only be opened. So when poor AMPA was opened, what happened? It got the conductance of the cations and there was a small EPSP. So what do I find here is a small EPSP. Fine. Now let us go to the next situation. This is my second situation when I started giving a tetany, when I started giving more number of impulses at successive intervals. So what happened here? As I gave more impulses, more amount of glutamate was released. More amount of glutamate was released. And these more of glutamate, they came and bound to the AMPAs. When they came and bound to the AMPAs, there was influx of sodium ions. When there was influx of sodium ions, since there was more of glutamate here, more of them went and bound to many of the AMPAs. Okay. So many of the AMPAs, when they opened up, there was influx of sodium ions. Ultimately, what was happening? There was an increase in the concentration of the positive ions. This increased presence of positive ions, it push the magnesium out. This is very, very important step we should remember. What the situation came? When we started giving the tetanic stimulus, when we started giving more number of impulses, more of neurotransmitters were released. More of neurotransmitters went and bound to AMPA receptors. More AMPAs were opened up. More influx of sodium ions. More influx of sodium ions increased the positivity, polarity, positivity of the cell inside. And these increased positivity has kind of pushed the magnesium out. So now what is happening to NMDA? NMDA is free of magnesium. Okay. So there is no magnesium which is blocking NMDA receptor. So what happens now when NMDA is free of magnesium? It is going to allow more amount of sodium as well as calcium. Look at this. Not only the sodium, since it's a bigger channel, it is going to allow more amount of sodium as well as calcium influx. And that is going to do what? That is going to increase this. So what am I getting? Okay, I got because of first impulse, then second impulse, then third impulse, then fourth. As the impulses were coming, then suddenly what do I find? The NMDA is free of magnesium ions. And so when it is free of magnesium ions, there was influx of calcium ions. And so what happened to this? It has increased. Okay. So initially there was summation effect, which has increased the height. Later, once the NMDA was opened up, there was increased influx of calcium ions. And so more of calcium got in and there was increase in the EPSP. So probably this EPSP, if it reaches the threshold level, it will give rise to the action potential. Now, next situation comes as what happens over here? What is not drawn here? Let me draw for you. Here, what will happen? AMPAs. So because of being more of a calcium coming here, this calcium ion, I'll tell you what happens to this. So post-tetanic, let me take you to the post-tetanic situation, okay? So what has happened initially? So this is the one, okay? Let me see here, okay. This is actually my post-tetanic situation. So magnesium is released. There is no blocking of NMDA receptors. More calcium has got in. Once this calcium gets in, there are so many things that this calcium can do. One such activity is that it can go and bind to calcium chem kinase protein. So it can go and bind itself with chem kinase protein, or you can say it stimulates the phosphorylation of chem kinase 1 and the chem kinase 2. And this chem kinase 2, what does it do? It causes the, what you called as, these receptors, they will go and they are going to bind externalization of the AMPA receptors. So more AMPA will go and bind to the postsynaptic end. So let me repeat once again. What has happened here? So we understood before there was no change. Once the glutamate came and bound to AMPA, only AMPA was opened up. There was influx of cation, and these influx of cation caused a small change in EPSP. Good enough. Then what happened during tetanic situation? When you gave the tetanic stimulation, more and more glutamate receptors, they came and bound to AMPA. There was more influx of sodium ions. This sodium, what it do? It went and caused the increase in the polarity of my what? The postsynaptic terminal. It has become more positive. And so what will happen? The magnesium is going to get released. So this is the situation, the magnesium which was blocking the NMDA receptor is no longer blocking it. So it will open up. Once it opens up, it will cause the entry or the influx of calcium ions. This increased influx of calcium ion to where? Please mind it. This is my postsynaptic end. This is not my presynaptic end. This is my postsynaptic end. This calcium is going to cause, a lot of biochemical reactions like phosphorylation of other enzymes, protein kinases, and that is going to cause the chem kinase 2 protein. And that is what is going to do is insertion of AMPA receptors. So more and more AMPA will be inserted on the postsynaptic end. So what does it mean? Whatever the neurotransmitter which is released my presynaptic is going to get taken up my AMPA. And how about my response here? This is going to be an increased response. So if I have to draw here, I will draw a bigger EPSP than the resting condition. Okay, this is what is called as long-term potentiation mechanism. So your postsynaptic end has become kind of more sensitive to the impulse which is coming from the presynaptic end. Okay, so this is what is the main mechanism for learning. So if you learn something, so I'll take up an example of your learning how to drive the bicycle. When you started in different stages. The first stage was you mounted onto the bicycle, you learned how to pedal it, how to handle it, now hold the handle and then pedaling. Okay, you were doing it. Then you kept on doing that job again and again. As you were doing it, you were giving more number of impulses from these presynaptic end. As you were giving more and more, what happened to your postsynaptic end? That has gone for tetanic response. And the post-tetanic response of this phase sign is that once you have learned in the stage 3, when you are now learned person, you kind of have learned how to ride a bicycle properly, even a small stimulus from the pre is going to give a good amount of response from the post. And why? Because now you have more number of AMPA receptors. And why they are more? Because they have been inserted due to the mechanism of what? The second messenger system, which is active once there is more availability of calcium ions at the postsynaptic end. Okay, so this is what you have to understand. I hope this is clear. Ankit, is it clear?
Yes, ma'am.
Is it clear?
Ma'am, I want to know about what is NMDA.
NMDA is another receptor. See, I can have multiple types of receptor. Okay, I can have multiple type of receptors for the same neurotransmitter. For glutamate, there are 2 types of receptors. Okay, there are AMPA receptors and NMDA receptors. If you want to know the full form, NMDA means N-methyl D aspartate receptor. Okay, it is a glutamate receptors, and this is also a type of ionotropic type of receptors. So what did I tell? There is ionotropic and metabotropic type of receptors. Okay, so and if you want to know what is the full form of AMPA, so I can tell you AMPA receptors. There is a big name, so usually we don't take the name of that, but for you I'm telling you. So the AMPA means alpha amino 3 hydroxy 5 methyl 4 isohexanol propionic acid. Okay, so we don't take such a big name. We just take them as AMPA-R. AMPA-R means AMPA receptors. NMDA means NMDAR means NMDAR receptors. N-methyl D aspartate receptors. Okay, so they both AMPA and NMDA are which type of receptors? Ionotropic type of receptors. And what are these ionotropic type of receptors? They do not involve the G-coupled protein mechanism. They themselves are like ion channels. So if any neurotransmitter comes and binds with them, so as you see here, let me take the laser and show you. As you see here, okay, if the neurotransmitter comes and binds to this, okay, this is going to cause the opening for NMDA. But what is so peculiar about NMDA and AMPA? AMPA is like small, simple receptor, very simple, honest receptor. Once a glutamate comes and binds, it will open itself. And so it will allow the influx of cations. But only problem with AMPA is a smaller channel. And how about NMDA? It is a bigger channel. So if it is a big channel with big pore, it will allow Not only the sodium, but also calcium ions also. Now, when can I have this NMDA opened up? We can have NMDA opened up only when there is an increase in the positivity of the cell to which this NMDA is attached. So when there is increase in the positivity, the blocking by the magnesium is released, okay, repelled. Magnesium is repelled out. So once this magnesium is repelled out, what do I have now? NMDA being open. And once it
Written NMBAR
this
hippocampus.
is NMDA
That,
receptor. R stands for receptor. Got me?
ma'am, that is written, ma'am, the word that is written to prior to hippocampus, that is, I think, miswritten, ma'am. NMBAR. I was asking
Oh,
about that.
this one. Yeah. Sorry, this is a typing error. Let me correct it here itself. Okay, you are talking about this. This is wrong. There was a typing mistake.
Yes,
It
ma'am.
is
I was
NMDAR. It is NMDA receptor. Okay, this is not B. This is D. Fine. Okay, then we'll continue. So we have understood about, as I was telling, the presynaptic inhibition, presynaptic facilitation. So presynaptic long-term potentiation, they has been best characterized in mosey fibers, in CA3 hippocampal synapses, as well as in parallel and Purkinje cell cerebral synapses. So where do I find such type of arrangements? This is the places that you can see them. You can find them in CA3 hippocampal fibers are the best studied fibers. Okay, so mosey fibers, CA3 hippocampal synapses, or you can find in cerebral synapses. Now, what is this? Let us start with the presynaptic long-term potentiation. This is another way of learning the long-term potentiation. So here, look up. Here, since it's a presynaptic long-term potentiation, what do I find? That these impulses, when they cause an increase in the calcium ions, these calcium ions which gets in, it kind of stimulates the another protein, adenylate cyclase. This adenylate cyclase is going to stimulate and cause the production of cyclic AMP. Cyclic AMP is again mainly going to cause the phosphorylation of protein kinase A. And this is going to do what? In turn, stimulate these enzymes, or you can say these proteins, RAP3A or RIM1 alpha. Please, you don't have to worry about them, but this is the mechanism through which it happens. Now, what are these going to do? They are going to cause more release of neurotransmitter. Okay, so calcium, which we were learning till now, that it was an anchoring protein and it was causing the release of neurotransmitter. Here, due to potentiation effect, there may be that more of impulses as they were coming here, more of calcium was available. And this calcium is not only going to cause as the anchoring protein and cause the release of neurotransmitter, it may also cause an increased neurotransmitter release via this cellular mechanism, biochemical mechanism of stimulating the enzymes. Okay, so such type of it, where with one impulse I could have, if it was a general or controlled impulse, with one impulse I would have got only this much. But because of my plasticity or more amount of impulses coming, what did happen? More amount of calcium was available at the presynaptic end. What has happened? This has caused more increased neurotransmitter release via this mechanism. Okay, so this is what is called as presynaptic long-term potentiation. Then let us come to NMDAR. Now, what we have learned till now was potentiation or synaptic facilitation. But there is another thing of plasticity where I can increase the number of synapses. I can also have a decrease in the number of synapses. There can be depression also. So it's like something, if you don't do anything repeatedly, you may even, if you don't use that thing repeatedly, or you don't make a habit of doing it repeatedly, you may forget about it. Your brain may start reducing the number of synapses. So this is how it forms a role in the memory. What is that? When you're reading anything more and more repeatedly, you're repeating it again and again, you're forming more number of synapses. You don't study it for 10 years, don't study it for 15 years, your memory will slowly fade away. And what has happened now? It is the number of synapses has got reduced. So that is what is called as synaptic depression. So LTD, let me take a pen and LTD, it stands for LTD. It stands for long-term depression. Okay. So as we had seen in the homosynaptic one, we have facilitation, we have depression. So long-term depression, what is it and what is the mechanism of it? We can tell about it. So we'll take an example of here where long-term depression is NMDA dependent long-term depression. And how is it triggered by? Let me tell you. So we started with, suppose there were impulses. Okay. So because of that, there was, again, I'm taking the glutamate as my receptors probably. So what has happened here? When the glutamate is released, it goes and binds to its AMPA receptors. Well, good enough. Then there was increase in the polarity of the postsynaptic end. Okay. So now let us go to this situation. Okay. This is as we have learned about the long-term potentiation, where we have seen that, after a tetanic, post-tetanic, what has happened, your post-synaptic terminal has become very reactive to any impulse coming from the presynaptic end, and it is responding at a very good way, at a very high response rate. How about long-term depression? Long-term depression is the opposite of long-term potentiation. And what has happened here? Long-term, and how does it happen? Let us study this. Here again, see, the glutamate is released. As the glutamate releases, it goes and binds to AMPA receptors. AMPAs are ion channels. They will allow the influx of sodium ions. More sodium ions will cause the polarity change. As there is a polarity change, the magnesium is repelled out from the NMDA receptors. If magnesium is repelled out, NMDA is now responsible for what? For opening. Now NMDA receptors are opened up and there will be an influx of calcium ions. So this happens if there is a moderate influx of calcium ions. This moderate influx of calcium ions, it had, in case of potentiation, it had an option to go and bind with CAM kinase 2 and cause the insertion of AMPA receptors and so cause a post long-term potentiation effect. But in case of LTD, this same calcium ions, what it does, it goes and binds with this calcineurin. It goes and binds with calcineurin. And calcineurin, it goes and stimulates the protein phosphatase 1. And what does protein phosphatase 1 do? It causes the internalization of the AMPA receptors. So how is it different from LTP? LTP was attaching the AMPA to the postsynaptic end, but during LTD, there is internalization of postsynaptic AMPA receptors. So this is important. Internalization of postsynaptic AMPA receptors. So if they are internalized, you don't have many AMPA being present at the postsynaptic end. And if they are not present, what about the response? I'll not get a stimulatory response, or you can say depolarization. I may get a reduced response. Okay. Now, that was one way of understanding LTD. There is one other way of understanding is when we take an example of metabotropic glutamate receptors. See here, this is a metabotropic glutamic receptors. So what did I say? AMPA and AMPA and NMDA were the inotropic receptors of glutamate. But glutamate also has a metabotropic receptors. And metabotropic receptors, what do? They are, what do they do is, see here, these metabo means They are kind of attached with G protein. They are coupled with G protein mechanism. Second messenger mechanism is involved, and a lot of other steps, biochemical steps are involved. And they will do, ultimately, this mechanism, although is not so clear, but what is very evident is the same thing. They cause the internalization of AMPAs. And this internalization of AMPA, it makes this postsynaptic end less reactive to the presynaptic stimulus. Okay, so that is one way of LTD. These are the mechanisms which have been explained in this review paper. Okay, so you won't find many of the mechanisms of LTD being present in most of the books. They just tell because LTP is by insertion of AMPA receptors. LTD, due to some mechanism, it causes the internalization of AMPA receptors. One such thing, which is quite well explained, is the one which is called as endocannabinoid receptors. Okay, this is Ankit, for your information. This is not that you have to study or learn it very well, but this is for your information, I can say. Okay, at your level, this may not be so important to know, but it is always best to know as what is the information still available. Because when you give a drug, where you're trying to, especially psychotic drug or psychoactive drugs, those drugs, they go and act onto the synapse basically by changing their potentiations. Okay, so they kind of, maintain at the neurotransmitter concentration, or they kind of increase the responses of your postsynaptic end to whatever the neurotransmitter is available. So it plays a very big important role there. Now, here is one example where we are going to read about the endocannabinoid receptors. So we have this is a metaboglutamic receptors. So what has happened? That whenever the neurotransmitters are released, these neurotransmitters, they will go and bind to their receptors, both the ionotropic as well as to the metabotropic receptors. These metabotropic receptors, they cause phosphorylation, and they cause the entry of the voltage, opening of the voltage gated calcium ions. So now calcium gets in. This calcium once gets in, it causes these cannabinoid receptors to be produced. Somehow they cause the release of these receptors. And these, what they do? They come out the postsynaptic end and they go and bind themselves to the presynaptic terminal. And here is where, by some mechanism, they inhibit the release of neurotransmitter. And this is how they cause the depression. So depression mainly occurs in two ways. One very popularly told is internalization of the AMPA receptors. And the other one, which has been explained in this paper, that is where these receptors, cannabinoid receptors, they are being formed, and these, they go and bind to the presynaptic end, and somehow they, they reduce the release of the neurotransmitter. So their mechanism is decreased presynaptic neurotransmitter released. So these are the two mechanisms which we find in the literature as what is the reason behind the long-term depression. Okay. So your synapse is kind of a place where I can have either inhibition or excitation, and it all depends as what exactly is the mechanism going on. Okay. So is there a modulatory presynaptic inhibition? Is there any modulatory presynaptic facilitation? Is there excitation? Is there inhibition? So one postsynaptic neuron can be under the influence of many factors. It can be inhibited also. It can be excited also. There can be modulatory neurons, which is going to modulate my presynaptic end. There can be modulatory neurons who are going to modulate my postsynaptic end also. And whole of their, mechanism is to either increase the synaptic strength or decrease the synaptic strength. You want to learn it more, you want to keep it in your memory, you want to, respond very quickly to any such stimulus or any stimulus which has been coming before, you go for strengthening of the synaptic synapses, strengthening of the synapses. So whole of these plasticity, you mold the synapses. Either you increase the synaptic integrations, kind of you synaptic connections, I can say, or you decrease it. Now, this is, look at this. We have learned a lot. What did we see? We have seen feed forward excitation. That is what you remember, that pre and post coming directly to each other. Then we had learned about lateral inhibition, which was by what? Rentschler cell inhibition. Okay, so we can modulate this lateral inhibition by increasing my synaptic strength. I can have more excitation if I want to increase my synaptic strength. I can do that. Then recurrent inhibition, you got, this is the Rentschler cell inhibitions, recurrent feed forward, feedback, then convergence, divergence. So we saw such type of connectivity, where we have seen this type of network being present, both excitatory and inhibitory in case of synapses. So this ends our synaptic plasticity. So very quickly, I'll realize it. Synaptic plasticity is a property of the synapses to change. It can change for a better potentiation, where, once you give the tetanic impulses, your responses becomes better and better. Or you can go for depression, where you give a impulse, but your response is decreased. Okay. Then there was another property called as reverberation. So with synaptic plasticity, I finished one of the property of synapse, that is synaptic facilitation, or that is where we have finished it. Now let us go to reverberation. What is this reverberation? Reverberation refers to the phenomena of passage of impulses from presynaptic neuron again and back to the presynaptic neuron to cause a continuous stimulation of presynaptic neuron. So what does this mean? Look at this diagram. That makes you more easier. This is my presynaptic neuron. It has given the impulse to my post one. Now somehow this post one is having extensions, the post neuron. After all, the post synaptic neuron is also a neuron. It may have axons, it may have dendrites. So some of them may go back again and stimulate the presynaptic neuron, which was stimulating the postsynaptic end. So your circuit is kind of reverberating. It's making a circus. It's going again and again. So now your activity of the pre is kind of modulated. It has become more active now. So this circuit is called as reverberation. And we can see such type of phenomena in case of synapses. So reverberation is a property of synapse. Then there is something called as after discharge. What is this after discharge? It's like hang on, it's like hang on. So you have a phenomena in which you have given a single instantaneous input, but you are not getting a single output, but you're getting a sustained output signals, a series of repetitive discharges. It's like as if you have stimulated a hyperactive person. So if you kind of go and stimulate a hyperactive person, he may not just stop in one conversation. Even after you have stopped giving him any impulse or information, he keeps on giving, blah, 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 blah. He keeps on telling it. So that phenomena is called as after discharge. You have given one single input, but you are getting many outputs, in very short period of time. So that is called as an after discharge. So that is, I think, important. Sometimes an event, it happens with us, especially with our emotions, that something has happened. You went and watched a movie in a cinema hall, and the movie had left so good impact on you that even after you have left the cinema hall, you're still kind of, pondering, thinking about the same movie, thinking about the same situations of the movie. So what are you trying to do? You are giving an after discharge. Okay. So with this, we end. We are at the end part of our properties of synapses. The last one is effect of acidosis and hypoxia. It means that your synapses are vulnerable. They their effect their property is affected, their activity is affected if there is accumulation of metabolites, if there is pH is decreased, or if there is hypoxia. So their responses is going to get decreased due to acidosis, metabolites, or hypoxia. Okay, so with this we come to an end to the properties of synapses. So whole of the properties of synapses we have covered in 2 lectures. With this, I stop here. Any questions you may ask me now.